This is episode 270, the Stem Cell Podcast, Microvascular Regeneration with Dr. Sara Vasconcelos. Hello, everybody. We are Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge and stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Sara Vasconcelos from University Health Network in Toronto. She's on the podcast to talk about her research on vascularization strategies to treat cardiovascular disease. We've also got our usual roundup of recent highlights and stem cell news. That'll be coming right up.
But first, we're very excited to meet you in a few weeks at ICCR 2024, happening July 10th to 13th in Hamburg, Germany. Be a part of the single most comprehensive event where best-in-class stem cell science and ample networking opportunities spark attendees, sponsors, and exhibitors to forge new relationships where ideas flourish. And also at the ICCR 2024, be sure to meet Dr. Nicole Quinn, host of the Lab Coats and Life podcast. She'll be there too. And also, just as a reminder, the first season of 8 full episodes has been released. So definitely check that out. Diving into the roundup for today, the first thing we're going to lead off with is a tech-oriented study in the gene editing realm. I mean, if you haven't figured it out by now, we are in the age of gene editing. It seems like every month we have a new type of editor that's coming out, usually from David Liu's lab over at MIT, but all around the world, right? And the thought is, the creation of these things is exciting, but it's the application that's even more exciting. And this is a paper in Science that's, focusing on one of those applications for in vivo editing of lung stem cells for durable gene correction in mice. This is coming from the lab of Daniel Swigert over there at the Department of Biochemistry in the Simmons Comprehensive Cancer Center at UT Southwestern. So this is an application of gene editing, like I alluded to, right? And we know all about in vivo genome correction, and especially since the age of CRISPR has come about in the last 10 years, this has really accelerated towards the clinic. There's a bunch of different companies that are using in vivo gene editing to target, a number of different approaches, ranging from the eye to the lung to the liver, and so on. And so this is an approach that has a lot of promise for generating durable, permanent disease cures. But one issue is effective editing of stem cells specifically, because you want to edit that stem cell population. And if you edit that population, that cure, quote unquote, will be propagated to the differentiated progeny, right? So here they're actually demonstrating that optimized lung-targeting lipid nanoparticles, or LNPs, are enabling really high levels of genome editing in lung stem cells. And it's a durable response. We're talking about persistent editing for multiple years in mice, which is a long time in mice, because mice don't live that long, right? This is an intravenously administered gene editing lipid nanoparticle. So it's an injection in activatable TD tomato mice, ultimately leading to greater than 70% lung stem cell editing. The key here is these lipid nanoparticles, like I said, are specifically targeting the lung. And this is really exciting because you can envision a scenario where you can modify these LNPs to target other portions of the body too, right? So you have something comparable in durable gene editing to another portion of the body, right? So this is a 70% lung stem cell editing in vivo, sustaining a TD tomato expression in greater than 80% of lung epithelial cells in these mice for 660 days. That's almost 2 years, right? And then they applied it to a disease model. They also addressed a cystic fibrosis model using a version of this editor, actually an adenine base editor, which is one of these next generation base editor point mutation editors, right? They introduced mRNA from that adenine base editor to mediate a greater than 95% edited lipid nanoparticle mediated edit of cystic fibrosis transmembrane conductance regulator, or CFTR, this really famous protein gene, whatever, that's implicated in cystic fibrosis. 95% DNA correction. That's phenomenal. This ultimately restored CFTR function, not just in the mouse model, but also in primary patient-derived bronchial epithelial cells, corrected intestinal organoids, and also corrected greater than 50% of lung stem cells in cystic fibrosis mice. So the big picture here is there are multiple model systems to validate this in vitro and in vivo editing approach using these lipid nanoparticles, these targeted lung-specific lipid nanoparticles, but that can also be modified in a modular fashion to target other cell types too. So this is, I think, really exciting. This is a tech-driven science paper. I think the application is, of course, really cool for editing cystic fibrosis, and that is one of the top diseases when it comes to genetic diseases that are amenable to gene editing and that are being targeted. But it's the adaptability of these lipid nanoparticles for targeting multiple tissue types, right? That's I think, the real beauty of this story is where else are they going to use these LNPs, right?
Yeah, the implications of this are vast, and it's, I think, encapsulates, well, just it's a good encapsulation of just the progress of science and tech. Um, in that, gene therapy has made such an amazing charge back from, I think, a low point decades ago. It was cystic fibrosis too, being a great symbol of the potential of gene therapy in the early days. I can remember Ron Crystal, who's a big deal in gene therapy, adeno specifically, who's at my institute here 20 years ago, around when I first joined. He was still moving along these therapies with the adeno-associated virus and nasal and something inhaled, but it just, the efficacy wasn't there. And it's taken, I think, the incorporation of seminal advances, transformative advances like CRISPR and all the base editors and David Liu products. But also, it's taken a lot of the lessons learned along the way from the failures in the early days of gene therapy, both regulatory as well as technical, and the incorporation of the other tech, like these lipid nanoparticles, all converging on something that's like, is this not ready for prime time? I mean, can this not be administered in early trials to human patients? I can't see why not. And it's just such an exciting time, right? Because as the tech is reaching fruition, as you said, there's so many unmet needs that can be addressed by this. I don't know. Maybe one thing is that you worry about outside the lung, off target, but if it's like a corrective mechanism, do you care? If it's correcting, the mutation in your liver as well, or wherever, you might worry about off target effect in another context. I don't know. Is there a reason, Arun, why this shouldn't be trialed in patients like in the next few years?
I mean, I don't think so. It seems like it makes a lot of sense to really accelerate this towards the clinic, and I think this is exactly what they're doing. And of course, you have to validate the off target. You always do with anything CRISPR related, but I mean that's the advantage of the adenine base editors is theoretically they are much less off target, right? Especially if you're just looking at a point mutation. And a lot of these diseases, cystic fibrosis, various other disorders, are all caused by point mutations in a single gene. And in that situation, the ABE, adenine base editor, is perfect. But as we know from David Liu's work and many other work in this area, there are newer approaches, even newer than base editors, like prime editors that could even modify bigger regions of the genome. And that's not to say that CRISPR-Cas9, like Cas9 itself, is dead. Cas9 is still the most commonly used endonuclease out there for CRISPR, but we know it has problems. We know it has big issues with off target. So the tech is continuously improving, not just in the CRISPR field, but in the delivery field too. You alluded to AAVs, which are still kind of the gold standard, but we have more directed approaches like these lipid nanoparticles now, right? So it's a great time for the tech.
Yes, absolutely. I mean, I can remember when I was like a young kid and they were pushing the Newton. You probably remember this because you're an infant yourself, but they had the Newton, which is like a handheld thing from Apple. And then they had the Palm Pilot. All these iterations failed, all of them. And then everyone thought it was dead. And there comes the iPhone. So it's just a matter of like all the pieces catching up with the idea, all the technical elements catching up with the idea. in order to culminate in like an actual effective product, in this case, a medical product. I'm still waiting for them to do something with the heads-up display. I mean, it seems like a lot of misfires with all these wearables, hurt your neck, and just no one has an interest, even though they cost 2 grand. But wait and see, Arun. It won't be long before wearing Ray-Ban sunglasses, sunglasses that keep us permanently in the internet, whatever they call it. Moving on, going from adult therapies to the very beginning of human development. And I'm not talking about embryo models here. This is a story that's actually based on critical observations of real human embryos, and I think underscores the need to continue this line of research with both human embryos and fetal tissue research. We've talked a lot on this podcast about the stories that come out and how critical they are. Here we're talking about really the first differentiation event, sort of, in a human embryo. We talked a few episodes back about a story from Magdalena Zernicka Goetz with the 2-cell stage being the first point of fate differentiation. Here we're talking about the trophectoderm, which is the first distinction of 2 different cell types in the embryo, the ICM and the trophectoderm. And of course, the trophectoderm is critical for development because that's the connection between the fetus and the mother, right? Or it's the precursors thereof, which is essential for the providing nutrients and removing waste. And as I said, the first differentiation event is when the outside cells of the morula stage embryo form trophectoderm, and the inner cell mass that will ultimately give rise to the epiblast, the embryo proper, and the hypoblast, which will form the yolk sac. Those are the 3 lineages that first emerge. And then the trophectoderm internalizes fluid to create the blastocele cavity, and the ICM is restricted to one side, or one pole of the blastocyst. And that pole is the trophectoderm that overlies the ICM is called the polar trophectoderm, and the rest of the trophectoderm, non-overlying the ICM, is the mural trophectoderm. In the mouse, the fully expanded blastocyst embryo, the trophectoderm is sustained as a single layer of epithelium. And the question is that also the case in human? It's hard to know because we don't have a lot of evidence from that early stage of development. It's not an experimentally tractable model, but there's a lot of ways in which human and mouse pre-implantation development and primary fate determination is similar. For example, you look at like YAP and hippo signaling, critical for trophectoderm formation. But also, there's notable differences between mouse and human pre-implantation development and trophectoderm formation even. CDX2, for example, drives trophectoderm differentiation in mouse, where GATA3 is thought to be the definitive marker of human trophectoderm, and CDX2 isn't really expressed in all trophectoderm cells. So that's a major difference from, like, in terms of the transcriptional hierarchy and inducers of fate. But also, like, at the gross level, the mouse embryo is encased in decidual tissue up until embryonic day 8, which in the mouse is like gastrulation has already started. You're getting, heart field formation, initial organ formation, neural folds. A lot has already happened by the time the decidua ultimately interfaces with the uterine epithelium and forms that interface, which is a long time. I didn't know that before reading this article. Amazing. Whereas in the human blastocyst, that polar trophectoderm overlying the ICM, that invades directly into the uterine wall, like right away, at 7-8 days post-fertilization, which is essentially as soon as the expanded blastocyst makes contact with the uterine epithelium. And currently, as you might expect in any human embryo model, little is known about trophectoderm expansion within the human blastocyst running up to that implantation at the uterine epithelium. But the fact that you have like such a accelerated attachment to the uterine wall, unlike the mouse where it's just swimming and the decidua is just hanging out before it interfaces with the epithelium post-gastrula or perigastrula, the urgency with which the human embryo attaches to the uterine wall would suggest that you got a lot of like proliferation and specialization of that polar trophectoderm in the run-up, because there's a lot of different cell types, the cytotrophoblasts and cystotrophoblasts, that are going to ultimately give rise to that maternal fetal interface. And it starts at that point. So, how do we look at this? Well, I mean, yes, human embryo model has kicked open the door to the black box, but also ART, assisted reproductive technologies, and IVF has afforded a lot of access to surplus human embryos that are available for donation for approved research protocols. And in this study, Dr. Jennifer Nichols from the Wellcome MRC Stem Cell Institute at Cambridge investigated how polar trophectoderm prepares to invade into the uterine wall by looking at supernumerary embryos that were donated for research, looking at days 5 to 7 in human blastocysts. And they used a lot of high-tech imaging methods, immunofluorescence of these rapidly isolated ICMs in order to distinguish ICM from trophectoderm. They also use blockade of RNA and protein synthesis and 3D reconstruction of immunostained embryos to ultimately show that there's multiple layers in the polar trophectoderm before contact and implantation with the uterine epithelium. They also show that the majority of the inner trophectoderm in the human blastocyst arises from existing outer cells. So there's no, and this is key, it's not like a conversion of residual ICM cells that are converting over to trophectoderm at this late phase. This is actually specialization and differentiation within the trophectoderm layer. And the ultimate upshot there is that this diversification and proliferation of these trophectoderm cells, it's still, while they're floating around, peri-implantation might be necessary in order to mediate this rapid invasion into uterine epithelium and to secure the developing human embryo to the uterus and initiate formation of the placenta. So this is a descriptive study with, I think, a lot of high-tech approach to really support this notion of a completely different, or not completely, but unique pathway toward trophectoderm specialization in the pre-implantation embryo. It's a DevCell paper. And I think it moves the field forward, not only for just basic understanding of how it works and how we might be able to use these findings to optimize in vitro growth of blastocyst embryos or transfer, but also lend some insight to as a benchmark to what we might be looking for in human embryo models that are proliferating at a rapid rate.
Yeah, I think everybody working in this field, and we've talked to a number of these folks working on early embryo models, I think they are so appreciative of this kind of work and establishing a benchmark, like you alluded to, for human development, early human development. And it's been a quote, black box for so long. The first thing I just have to say is you just have to admire the folks doing this kind of work, because as you very well know, it's so hard to obtain these embryos. Even if this is a purely observational study, it is incredibly important and very difficult to do. They even said, the human embryos left over from assisted, conception programs exhibit considerable variability in cell number, blah, blah, blah. There's a lot of heterogeneity, even from embryo to embryo, as you might expect at the early stage. So these things are so precious. And they were able to use them to the full potential. And certainly, like what you're alluding to, this is going to be so important for establishing that benchmark for how well the early embryo models are actually developing and how true to the real thing they are. And we covered a paper a while back using endometrial organoids in combination, I believe, with the blastoids that are being developed by the Rowan lab to look at implantation and those mechanisms and so on. And this is just going to further solidify and further, like I said, serve as a benchmark to study those, the newer generation of tech that's attempting to emulate these early mechanisms of development. So really cool study just from the observational perspective, but with a lot of implications for developing better versions of these embryo models, right?
Absolutely. And circling back to your point about the diversity of embryo morphology in the context of IVF, yeah, I mean, that's a huge point. We have so few parameters to examine when trying to diagnose the health or likelihood of success of embryo implantation. And this is a major element of that scorecard, I think, that should be added. I don't know now that we are currently, it's all light microscopy, although time lapse, where effectively in most large practices you have a movie of every embryo developing from single cell zygote to blastocyst stages in most cases, and the dynamics there is big with the AI analyzing how they grow. But given a method to actually evaluate how many layers there are in the trophoblast or what the degree of proliferation is in the trophoblast, which I don't think are out of the question, I think this could be a major addition to the scorecard for identifying blastocysts that have the greatest chance to implant and lead to a successful pregnancy, or on the flip side, to diagnose patients or embryos that are facing repeated implantation failure, which can be so frustrating, especially when you don't have a reason, you don't have an explanation for the patient. So, despite or in addition to the amazing tool or application here in terms of benchmarking, this is also maybe a big add for clinical assessment.
Yeah, I mean, human development is just so fickle. It's crazy how different we are in terms of developmental potential from other mammalian species, right? And certainly, birth rates are going down, so that's why IVF requirements are increasing, and the advantage of IVS is going up, and I think the percentage of folks who are receiving it is going up. And I forget the number, but a significant portion of human embryos are lost prior to gastrulation, right? And these sort of studies are going to help inform IVF, like what you're alluding to. So all great things, and that's completely independent of the embryo models, which we talk about every single episode, right? So moving on to something a little different, we're talking about not development, but regeneration. I suppose two sides of the same coin, right? This is regeneration in the liver, which has this astounding regenerative capacity in contrast to other organs like the heart, which don't regenerate very well. This is a science paper coming from Prashant Mitra. And this is a really cool study looking at the metabolism of hepatocytes specifically and how that influences the process of liver regeneration. Apparently, there are certain subclasses, metabolic subclasses of hepatocytes that are better for liver regeneration than others. And it all kind of depends on this node of mitochondrial health. So, in some aspects, the study is kind of straightforward. The bottom line, if I could summarize it in a sentence, is that hepatocytes with a functional electron transport chain are better able to contribute to liver regeneration, which is sort of obvious, right? But there's more to it than that, and they really teased it apart pretty well. So we know that the electron transport chain dysfunction is observed in a bunch of different human diseases, including in the setting of liver diseases as well, especially metabolism-associated liver diseases. And during this process of liver regeneration, you have to have the proliferating hepatocytes to do the work. They actually compete with each other. And there are cells, hepatocytes with high fitness that outcompete those that aren't as good, which is kind of what you want. You want the healthy cells to restore the liver in its whole, right? And there's some recent studies that have shown that mitochondrial electron transport chain can actually influence stem cell behavior. It's not really known, though, whether the ETC, electron transport chain, can influence hepatocyte proliferation specifically after liver injury. I mean, we're talking about like various injuries to liver, alcohol mediated injuries, and so on. And this, like I said, is really important for restoring the liver to its proper potential, and ideally to the way it was before the injury, right? So here they use really cool metabolite profiling of the mitochondria in hepatocytes in the liver, and also different isotope tracing techniques in mice to actually investigate the metabolic response in hepatocytes under homeostasis, standard conditions, but also in regenerative conditions. And then they use a bunch of other mouse models targeting the mitochondrial electron transport chain to tease apart what are the contributions of specific electron transport chain complexes. And as a reminder, there's like 1, 2, 3, 4, and 5. They tease them all apart. And they wanted to identify what's the contribution of each of those subtypes, those complexes, to the regeneration process in the liver. So ultimately, they were able to show that the mouse hepatocytes required a functional electron transport chain, kind of sort of obvious, to proliferate and actually compete with the wild type hepatocytes during the liver regeneration. So these things have to be healthy to restore proper liver function. And it's the healthy hepatocytes with a good electron transport chain that are going to be the ones that are outcompeting the other ones, which is what you want, right? But to confirm that, in the absence of the electron transport chain, mouse livers accumulated stuff that is not great, which is like fatty acid species, steatosis of the liver, not good stuff, right? Another really cool element of this paper was another observation that they made during that process of regeneration, where there's actually a transdifferentiation of the cholangiocytes, another cell type found in the liver, into hepatocytes, while those electron transport chain deficient hepatocytes were stimulated for regeneration. So it's almost like a compensatory effect that's going on. If you have bad hepatocytes when it comes to their electron transport chain, there are other cells that will turn into good hepatocytes to ultimately make the healthy liver, right? And then they did a bunch of other metabolic tracing, showing that the wild type livers rely on mobilization and oxidation of peripheral fat stores to maintain acetyl-CoA during proliferation, like a really deep dive into metabolism. And ultimately, they arrived at this a single node regulated by PDK4, which is a negative regulator of pyruvate oxidation. And this particular enzyme is driving that metabolic inflexibility, where only the healthy electron transport chain, healthy hepatocytes are the ones that are able to contribute to the restoration of the full liver. And it's that specific enzyme, the PDK4, that's regulating the healthy hepatocytes there. So it's a really deep metabolic study, and I'm not giving it complete justice, but I thought it was just really cool how they teased apart a sort of obvious observation that you need healthy hepatocytes that are electron transport chain sufficient and proficient to do the deed and actually restore the liver to its full potential after regeneration. They really dove into it, figured out this one node in PDK4 that's regulating this process. And I think it has potentially a lot of implications for making sure the liver gets back to where it's supposed to be after, say, alcohol-mediated injury and so on. So really cool basic science study in the most regenerative organ in the body. Although perhaps the skin has something to say about that too, right?
Yeah, I was just thinking, the liver is such a great playground for trying to understand all these unique mechanisms of regeneration over a lifespan. It's really impressive, the scale and breadth of regenerative mechanisms that exist there. But yeah, is it relevant to any other tissue? Because it's so unique. The liver has been tuned over the course of eons to have these, I would say singular mechanisms for regeneration. But I raise the question: is there similar mechanisms in place in other tissues that are highly regenerative? Might be worth a look. But for me, as you said, I mean, it seems pretty straightforward. But behind that, I would say, as you were describing, the depth to which they went here, and conceptually, I think it was interesting, and I would say anti-dogmatic, maybe. Just the idea that this metabolic inflexibility is the key, like creating this bottleneck where only the capable or the healthy, I guess you would say, hepatocytes are able to contribute to regeneration. I think it's maybe also kind of obvious, but as opposed to our general appreciation of metabolism, where we understand that cells in different environments or under different stresses may need to exhibit some metabolic flexibility. Like it's a key, the adaptation. It's like the key to everything, right? But in this case, the overall benefit to the organ and the organism in this case is predicated on this notion of metabolic inflexibility, which I like as an idea. Is that sometimes you got to, tighten the reins in order to keep the organ on course, as opposed to maybe enabling some of these maladaptive cells to come into play and undermine the health of the liver, and thereby undermine the health of the whole organism. So I think an important validation of our intuition, while also advancing our conceptual understanding of how these things work.
Yeah, I mean, ideally, of course, you always want the healthy cells to drive the regenerative process, but that is a generalization, and sometimes that ideal doesn't always manifest because what is regeneration gone wrong? And you can perhaps think of it this way: cancer, right? And cancer is a situation when, of course, it's the not healthy cells, the hyperproliferative, incorrect cells that are ultimately taking over the population. So we always hope that things work the way they should, but the human body and the body itself is it's not perfect, unfortunately.
Yes, not to mention, I mean, cancer, not to mention fibrosis, which is the other major impediment to healthy aging. And it's a good segue to my next story. Also, a good segue to our guest, who has a tremendous investment in vascular biology and microvascular vasculoplasty therapies to improve vascular perfusion in tissues. And it's a really important unmet need. Peripheral artery disease affects 20% of adults over 60. And by the time you get to 80, about half of you are going to have peripheral artery disease. I don't know about, I'll probably get it. Arun, you'll probably be okay. You're a healthy guy. But it's severe. I mean, the disease symptoms ranging from pain only on walking to chronic limb-threatening ischemia, which is the extreme case. It manifests as intractable pain at rest and often accompanied by ulceration, gangrene. And this is a real hit to me. The patients having quality of life similar to those with terminal cancer. So this is like, for that large a segment of the population, that's a pretty huge unmet need. Um, and There's like 500,000 new diagnoses of this chronic limb-threatening ischemia in the U.S. alone every year. More than half of these patients are going to die within 5 years, and about 60% or more of those um, who are severely affected are going to require an amputation within a year of getting their diagnosis. Needless to say, all these treatments are tremendously costly, and as I said, the quality of life is significantly diminished for these patients. So, you know, I'm sure Sarah will tell you, there's been a lot of therapies proposed, mostly based on this idea of proangiogenic stimulus. And then despite a lot of advance and positive data in preclinical studies and animal models, when you gone through with the placebo-controlled randomized studies using either proangiogenic or proarteriogenic cytokines, or mixed populations of peripheral blood, or bone marrow-derived mononuclear cells, none of those have shown any clinically meaningful efficacy. Um, and even when you look at these proangiogenic or proarteriogenic cell types, namely these peripheral blood mononuclear cells, there's not much understanding of their mechanism of action. And even the cell types that actually are the business end of that proangiogenic effect, they're not really well described. What we do know is that it's a small percentage of those cells that you inject are actually doing anything positive. There may be even cells that are negative. They're causing inflammatory stimulus that is not proangiogenic. Um, anyway, what we're talking about in terms of the cell-based origin of these proangiogenic signals, it's monocytes. They're a subpopulation of mononuclear cells, as CD14 positive cells. They're known to have an important role in innate and adaptive immunity, but they're also have been shown to have these tissue remodeling properties. They're mobilized in response to ischemia. They accelerate endothelialization, re-endothelialization. They promote limb salvage in these murine models of hind limb ischemia. And monocytes can be segregated further into a classical, so a CD14 positive, but CD16 negative subset that modulates inflammatory effects of monocytes. And then there's this non-classical population, which is CD14 positive or low, but CD16 positive. And those have been shown to have a proangiogenic profile. So, owing to that capacity, a group led by Alberto Smith and Bijan Modarai, sorry about the mispronunciation if there was one, from King's College in London, they set out to investigate and to really uh, parse out and deconstruct the influence of these pro-angiogenic non-classical monocytes in a pretty simple way. They looked first in patients that had chronic limb-threatening ischemia and found, as you might expect, that the level of these CD16 positive proangiogenic arteriogenic monocytes was increased. And when they compare them to the CD16 negative or inflammatory classical monocytes, they showed that in vitro, the CD16 positive cells had large vessel remodeling properties. They also showed when they injected those cells, the 16 positive ones, into a hind limb ischemia model, that they were able to mitigate, or they were able to salvage the limbs in those patients. They show that these PAMs, as they call them, 16 positive cells, also secrete a lot of angiogenic and arteriogenic factors, including VEGF-A, heparin binding epidermal growth factor. And here's the kicker. This is a science translational medicine story. They actually put it into people, this first in human. It was a single arm cohort study. So, I mean, minimal patients, but they had 5 patients, I think, that they injected these cells in. They were labeled with this indium-11 isotope, indium-11 labeled topoline. I don't know what that is, but it's a way that they could track these cells. And they showed that at multiple time points, 2, 24, and 72 hours, they looked, and at 72 hours, 25% of the injected cells were still there, which is significant given that you typically expect a larger degree of washout. So it seems to be like there's some, I wouldn't call it engraftment, but adherence within the site, or transient engraftment at the least, out to 72 hours. And when they looked at viability amongst those, they showed that 80% were viable. They were able to do that because and this is again, N equals 1. They had one patient that was scheduled for amputation. So 10 days after the injection, they took a biopsy of that amputated limb relative to the non-injected muscle in that limb, and showed that there was a 80% viability, and also 2.5x the size of the vessels in there and 1.5x the number. So I would say a measured improvement, a step in the right direction. Not going to be reversing the ischemia maybe tomorrow, but I think the first step here is key, which is isolating the cells, showing that a significant percentage. I mean, in the past, you'd inject, let's say, 100 cells, and there'd be 2 that were left over because so few of them were actually doing anything to begin with. So here, showing that you can really inject the target population that sticks in there and has an effect, I think is a major step in the right direction. And I think that the technology can only be refined. And I do think, even though the improvement here is modest, that this perhaps justifies, especially in this patient population, where what are we looking at? It's autologous cells in a limb that you were going to cut off most likely anyway. So I think it justifies a clinical trial in humans of a larger scale and scope. So I think this is exciting. I'm someone who came into this field as using the hind limb ischemia model, and a lot of the approaches to date have fallen flat. So I'm very excited to see this progress. Arun, what's your take?
I'm actually shocked that this is not a bigger paper. I mean, I love science translational medicine first and foremost. So, like, I've published in the journal, and they have such cool translationally oriented stories there. But you were talking about the first half of the paper, and I was like, wow, this is, really exciting stuff with the PAMs, right? The monocytes. But then you talked about the second half where you actually had this first in human single cohort study where they had positive results. I mean, that's a difficult study to do. There's a lot here. And like what you're saying, peripheral artery disease is a huge medical burden. And so the potential impact of something like this is tremendous. We're talking about millions and millions of folks around the world dealing with these problems. I'm actually shocked this is not a bigger paper. Are you?
Well, I wonder what course it took because, I don't know, maybe the reviewers saw the mouse data and wanted some human, but I doubt it because you got to prepare years in advance to introduce these things into patients, I would expect. So I'm not sure what held this back, but I would venture that maybe the limited N is what kept it out of maybe nature medicine. But science trans is nothing to sneeze at, right? Science translational medicine, and I think it's an appropriate forum because you see a lot of nature medicine has gone so clinical, not in a bad way, but it's I think read primarily by MD PhDs who are clinical scientists, 70/30 bona fide, actually in practice. Whereas I think this is a real science geared story that could be applied now in all kinds of models, and is I think a first step towards something that, as you alluded to, will probably be published in a much higher, I wouldn't say higher, but in a different forum, with a greater group of patients, and really measuring benefit in terms of recovery or avoiding amputation. Maybe that was it. They ended up cutting this limb off anyway. You would like to see that you could administer this at an earlier stage or at a less grave stage of the peripheral artery disease and actually cause recovery or reduce the ulceration and gangrene. I'm guessing that that's where they're taking this next step with a larger scale phase 1 trial. Well, we'll have to talk about all the many approaches to saving our limbs with Dr. Vasconcelos. But before we get there, got a quick message from STEMCELL Technologies. Do you work with human pluripotent stem cell-derived cardiomyocytes? Use STEMCELL STEMDiff cardiomyocyte media and supplements to differentiate, enrich, expand, and cryopreserve functional HPSC-derived cardiomyocytes. STEMDiff cardiomyocyte media is compatible with human embryonic stem cells and induced pluripotent stem cells. And the resulting cardiomyocytes can be used for disease modeling, drug discovery, and cardiotoxicity screening. Learn more at www.stemcell.com/stemdiff-cardio. I can see Arun punching those letters into his keyboard as we speak. He loves the STEMDiff. Now on to our interview. All right, everybody. Today with us, we have Dr. Sara Vasconcelos, who is senior scientist and John Kitson McIvor Endowed Chair in Diabetes Research and University Health Network Associate Professor at the Institute of Biomedical Engineering and the University of Toronto. Dr. Vasconcelos' lab is interested in uncovering the principles that govern vascularization during organ regeneration for application in regenerative medicine. They study microvascular regeneration in cardiovascular diseases and in diabetes, and are working to develop personalized patient-derived organ chip models for drug screening and other applications. Sara, thank you so much for joining us on the show today.
Thank you guys for having me.
Yeah, thanks for being here. Always great to chat with a fellow cardiac biologist. I mean, like Dalen mentioned, your lab focuses on all things cardiovascular, right? And utilizing developing tissue engineering approaches to address cardiovascular problems, various basic science applications in that area. It's an area of interest that's close to my heart too, pun intended. I mean, different projects in your lab focus on microvascular regeneration, cardiac regeneration with stem cell cardiomyocyte, or organ on chip applications, and I think we'll touch on each of these a little bit during our conversation. But can we just start off by giving us an overview in your words of what your lab is working on right now, and maybe what inspired you to start working in this area in the first place?
So I think you did a very good job of kind of covering what we do, and it's a pretty broad, right? But basically, our goal is to work on the regenerative medicine field for cardiovascular disease and diabetes. And a big unmet needs, particularly when it comes to cell-based regeneration, is having a functional vasculature that goes with the cells, right? So to enable these cells to survive and to function. So that's one of the main goals in the lab. And it falls under this kind of two umbrellas, the more in vivo stuff where we do cell transplantations and grow vessels in vivo, but also the in vitro stuff where we create these models to study tissue homeostasis, disease development, and screen for drugs with the end goal, again, of this regeneration, right? So it's tied together. And in terms of how I kind of end up and my passion for this, I really am a trained vascular biologist. So undergrad, master's, PhD, that was the core of what I did. And then slowly adding up throughout my training and PhD and postdoc, tissue engineering aspects of it, as well as stem cell derived cardiomyocytes. So that's kind of how it all came together in terms of this cardiovascular regeneration.
Yeah, and I like your answer there because I think it really captures how vascular biology and this unmet need that you described is really at the center. It's like the convergence of all these worlds. There's the modeling, there's the functional, tissue regeneration element, and also in terms of approach, it's at the nexus of the kind of engineering as well as biological insight and understanding and inquiry. And it takes, all those pieces, I think. We talk a lot about this on the show about, like, do you build the thing? Can you construct it, or do you grow the thing? And I think that's a question that we're still struggling to answer in terms of translation. But as you said, it's really about one of the major endpoints is revascularizing tissue for regenerative purposes. And I think, as much as Arun said, as a cardiac biologist, I think he really appreciates your work and the relevance of the vasculature, because the heart is, literally and figuratively central to life and blood flow, everything that follows from it. But I think anyone who looks down a microscope as a developmental biologist, seeing something grow from, a zygote or whatever stage up to a larger organism, you can appreciate the centrality of the vasculature, because whatever tissue or organ you care about or organism, scale requires plumbing, right? Growth, you need the conduit to distribute the oxygen and nutrients and to get rid of all the toxic metabolites, et cetera. We all know that story and that challenge. And a lot of your published work to date has utilized this idea of ready-made vessels for improving vascularization of grafts. As someone who worked a lot with endothelial cells derived from pluripotent stem cells, we talk about that a lot on the show. It's Stem Cell Show. I recall a major rationale for that approach was we'll have an inexhaustible source of endothelial cells for, like, these vasculoplastic approaches in regenerative medicine, or as an organ assist, or to co-transplant with whatever tissue. And there were a lot of challenges there. But I would guess that a scalable source of these ready-made microvessels is also a challenge, right? Or maybe it's not. I mean, you tell me, what do you see as the most practical solution long-term for vascular assist and tissue grafting for regenerative purposes? Like, I know you're on the microvessels now, but do you think that's going to be sustainable long-term? You think ultimately we're going to have to do a cultured cell? To talk about it.
Oh, that's a very complex question. So let's tackle it into smaller pieces. So we use these microvessels that are isolated from fat. So in theory, we think about this as recycling. You're taking blood vessels from an organ that you do not need to survive and putting it at a place, a ischemic environment, like in the heart or etc., that can help with rebuilding vessels. And it seems to be a self-contained kind of like organism. It knows what to do, it grows, it connects, and it expands, and it becomes perfused and helps with cell transplantation and etc. So in terms of potential for application in the clinic, the potential is it's quite high because you can always do a small liposuction. It's even a procedure now, right? It's not even a surgery. And then take the fat, we can store them, can be freezed, and you can use this later on, hopefully from the same person. But with that also comes a challenge that, if you have cardiovascular disease, you had an MI, a stroke, or something like that, or diabetes where you need to replace those cells, you likely have some vascular deterioration, right? So we are not sure at this point which patient's cohort would benefit most from this, right? So if you think about diabetes, you think about microvascular damage. So those might be, a more challenging cohort to treat, as opposed to someone that has atherosclerosis that mainly impacts the larger vessels. So the way we're thinking about this is, I think we can use those in the clinic, and that's something we're considering and working with. The good thing about being in a hospital is that you can work with the surgeons and kind of come up with strategies to advance that. But the other part of it comes from having the source, right? As you said, if you have a source of cells that you can use unlimited to create these vessels, that would be really interesting. And also from a regulatory perspective, right? So if you're approving a transplantation of beta cells or cardiomyocytes, you want to ideally have this isogenic product that you can use, and then will make your life much easier in terms of, having applying for approval from the FDA or here in Canada, right? Or other places. So based on that, and with all our curiosity about how really these microvessels work, we have started now and looking into really in depth, what is it about the microvessels that make them work so well? How they have this program, and can we recapitulate that with stem cell derived vascular cells in general? Because the microvessels we use are not just the endothelial cells, right? They have other cell types, mural cells, and etc., that we think play a key role, right? So now we're investigating, is it a particular cell type, or is it the combination of the cells, or, likely it's multiple factors that are leading to this functionality in these microvessels.
I think that's a great answer, and I'm sure Daylon would agree too. But on that topic of using stem cell derived vessels, I mean, this is something we talk about a lot here on the show, and I know Daylon has very strong opinions on this. No two endothelial cells derived from iPSCs are the same, right? I mean, it's a mixed bag, and it's a reflection of the heterogeneity in the body as well. And we covered a paper a while back where they described like 200 different independent, unique endothelial cell types that are found in the body. And I think we're just scratching the surface of what we can actually make from iPSCs in terms of, vascular cell types, right? But outside of the cell therapy applications, I actually also wanted to talk about some of the tissue engineering applications that your lab has been working on for disease modeling and so forth. Like, for example, this cardiac fibrosis on a chip system, right? And I'm somebody who's done some heart on chip work as well, so I can appreciate the level of difficulty in getting this complex system off the ground. You got to deal with the cell biology part of it, like we're talking about, right? Getting these cells to function, to adhere in this chip, I'm sure, as well as the engineering side of it and picking up which material you're going to use for the chip, whether it's PDMS or some other substrate, right? Figuring out the right orientation for these cells. So tell us more about this engineering oriented work and maybe where you want to go next with it.
So that's a very interesting question. And especially depending on the audience. So if you talk to biologists, they're very skeptical. Oh my gosh, this is definitely not a heart. Well, we never said it is a heart. But how is it any different than culturing your HUVACs on a super solid plate, right? It is not. It is a tool that you can use. And if you know what you can measure and what you can use it for, if you have characterized it well, you know that you can, use it to identify particular things that you have characterized the tissue for. So that's the number one thing. And particularly for trainees and people joining the field, there's this, oh, it's a heart, and it mimics certain aspects of the heart, right? And then what we tried to do with that cardiac fibrosis on a chip was very simple model of trying to just put in an insult that is associated with fibrosis and seeing whether that would replicate what we see in the clinic in terms of fibrosis, right? And we characterize it heavily and et cetera. But, I find that for us to be able to do all these different things, it's very nice to work with people from different backgrounds. So we have engineers, we have biologists in the lab with different types of training that can help us overcome some of these limitations. If we're bringing different cell types, you have to really look at the cell culture media. Sometimes you can't, it's not going to work. And an example of that is our work on diabetes. We're trying to get the microvessels in vitro with the differentiating pancreatic progenitors. And a requirement for the pancreatic progenitors was a notch inhibitor, which is totally counter to growing vessels, right? So that, those limitations, it helps when you have, this interdisciplinary team that can think outside the box and try to address some of these limitations. So for that paper, it was really very simple, and we insulted the tissue, like activated the fibroblasts, and let it just take its course, right? And then we saw differences in tension, basically passive tension, right? Which is usually what we do. But what was nice that because there's a lot of fibrosis tissues out there. And at the time, there were already some, and they were all mostly just the fibroblasts, right? So I think what we did really was when we put the cardiomyocytes, we could, in real time, assess, function and see, how different drugs would affect it and whatnot. So I think that's what, is cool about that. Not to mention that, I don't know about you, but every time I look at the beating cardiomyocyte in the plate, I'm still like super excited about it. What's really a disappointing for me when I brought my 8-year-old to the lab and I was like, look at this, it's so cool. She's like, okay.
Oh, man.
She wasn't impressed at all. It's like, okay, maybe you need to know more of what we're doing.
Yeah, we talk about that a lot. That never gets old. And it's kind of what draws, I think, a lot of us in for life is the first time you see that as a stem cell biologist in particular, where you're like, wow, life, the origins of life happening in a dish. Amazing. Don't worry, the kid will come around once a little more context perhaps necessary. But yeah, I like what you're talking about there because I've always lamented, and my former mentor, Shaheen Rafi, is always, he gets up and the first thing he does is yell at everybody about how endothelium is not just plumbing, boss. And he's right. And he's also illustrated how instructive it is, the angiocrine hypothesis that I think has borne out in a lot of different vascular beds. And what you're describing there, disease modeling, I don't think endothelium gets as much attention as it merits, considering that diabetes, cardiovascular disease are like the twin scourges of our, first world culture. So I get it, why it really draws interest for your lab and others. But you alluded to it there, the crosstalk between endothelium and the parenchymal cells in any tissue organ. That's really critical to that organ-specific identity. And there, in that case, you had to get the pancreatic progenitors there, and that introduces all kinds of technical obstacles. Could you just elaborate a bit on that? I mean, I don't want you to try predicting the future here, but like, do we need to get to a point where you're actually like intercalating parenchymal cells or organ-specific cells with endothelium, like in the same architecture as the tissue, perfused at the microcapillary level? I mean, it seems like a big ask. Or can we reconstruct that interrelationship with these engineering approaches like you were kind of alluding to there, but like, is that feasible, or are the technical barriers there in terms of different, media requirements, for example, do you think they're going to be really difficult to surmount?
Well, there's work out there that has put endothelial cells with cardiomyocytes, right? Arun will know, and other people that have also done some of that. I don't think you need to recapitulate 100% the architecture and the details. It's really, again, about the questions you're trying to answer, right? And whether that those endothelial cells will be instructing the other cell types, right? It's been shown that endothelial cells are important for cardiomyocyte maturation. So, talking about that signaling, right, from the endothelial cells into the cardiomyocytes. So I think it's really important. And I think people don't appreciate because, and I'm a bit biased, but they don't really appreciate how the endothelium is an organ on its own, right, a vascular system. But it's also extremely different within the different organs, right? And we knew that, we've always known that some are fenestrated, some are not. But as you mentioned yourself, with the single cell RNA-seq fever, now we know there's all cells are different. No cell is the same, right? So when you're talking about organ-specific systems, how do you achieve that? Right. And there's going to be different levels. So like in vitro, it's likely going to be complex for certain reasons. So more like architecture and the media choice and etc. And in vivo, we haven't even answered that question yet, right? Do we need tissue-specific endothelial cells for regeneration? And that's something we've been working on because since we've shown that our microvessels really persist and form the bulk of the vessels in the graft, it gives us an opportunity to ask those questions. Okay, so if they stick around and they stick around long term, we've shown in diabetes that up to 3.5 months later, 70% of the vascular cells in the implants come from the donor vessels. Can we now ask those questions? Are they becoming more like pancreatic cells or not? And that's kind of how we started doing some of the single cell work because we took the data, we wanted to look at it, and the question is simple, but it's very important, right? Because people are already differentiating tissue-specific endothelial cells from pluripotent stem cells. So that's great, I think, for in vitro applications. But do we need it in vivo? We may not. So from a vascular perspective, I think they're plastic, and up to some point, they can take up some of those characteristics. So we're really excited to be able to compare. Okay, let's look at that level and see how our cells pre and post implantation compare to the cells, endothelial cells in the organ, the native ones. But we came across an issue that pancreas was really overlooked, right? If you look at those maps or those atlases, the mouse atlas, etc., you don't see a lot of pancreatic, there's actually no pancreatic cells. And there's more like individual work done in pancreas that has some endothelial cells. So we had to kind of come up with an pancreatic endothelial cell signature to be able then to rank our cells that we implant. So we're doing that now. And we mentioned that the bioRxiv paper that we have that was done by a talented PhD student in the lab, really analyzing that and validating it in an independent model, right, in an independent data set, to make sure that whatever we got, in terms of gene would be representative, and then we'll be able to score ourselves on that. We haven't done the scoring yet, so I'll keep you posted. We're finishing. We wanted to have more N, so more endothelial cells to be able to have a more robust statistical analysis. So that should be coming. Really excited about that because I think that they're gonna, really take on this more pancreatic phenotype when they're transplanted with the pancreatic cells, but we don't know.
Yeah, I mean the benchmarks I think are really critical, and yeah, sometimes you got to go out there and create your own. But we've been talking about that also on the show a lot, about all these standards that are putting out all these atlases, the endothelial atlas and human and mouse being one of them. But it's an evolving data set, and I think the key is to have the good work done on the front end. Getting back to like the kind of regenerative therapeutic element. I was reviewing some of your very diverse and generous funding. Congratulations on that. From your lab's website, was intrigued by one of the projects that was related to microvessel-based heart repair in a large animal model. I mean, I think it's one of the earliest day studies. It was like, we got to see how we can get vessels into the heart for heart repair, but it's made a lot of progress, but there's still a long way to go there, I think. And the large animal model part of that, I think, really intrigued me. So much, literally decades at this point, the proof of principle in the stem cell field based on mouse models, obvious reasons for that. But of course, everything's got to go through a large animal or non-human primate model before it's ready for prime time, right? And it's about scale, obviously, right? But, of course, bigger organs, bigger infarct, I guess, greater mass of perfusion you're going to need, more cells, right? But is it just a matter of numbers? I'm sure there's more nuance there. What are some other critical endpoints and or experimental parameters that you need to consider that really aren't that critical in the mouse studies? What's different about these large animal models that makes it either more challenging or some fun facts about the large animal models and the challenges there?
Yeah, it's actually been a very steep learning curve for me because we didn't do it before, right? And all of a sudden we're doing pig work in the heart and it's infarction, and you need to be on top of all these different things, right? But it really is the gold standard for works in the heart, right? Because the size of the heart, the shape, the rhythm is the same. And that was really brought out the fact that, the stem cell derived cardiomyocytes are so immature that they actually drive arrhythmias in these models. And we can't pick that up in the small animal models, right? So that's a very important thing. And what we're looking now is, because we have some evidence from histology that, our microvessels secrete things that, cause hypertrophy in these immature cardiomyocytes, suggesting that potentially there's some maturation there. This is the perfect model to try to assess some of those arrhythmias and see if it's affecting it in a beneficial way or not, right? The other thing about the pig is scalability, as you mentioned, right? There's only a certain amount of material we can inject in a very small rodent heart. In the pig, we can really do some of those dose response tests, right? So we use this concentration of microvessels, the ratio of microvessels to cardiomyocytes, which was very low. It was about 200,000 cells in the shape of microvessels to 1 million cardiomyocytes. Now, moving forward in the pig, we can do different doses, right? We have this very large ventricle that we can do 10 to 13 injections. So we can really test, okay, now if we put a lot more vessels, is it really going to improve? But the biggest issue for those studies is the immunosuppression, right? You have to very heavily immunosuppress these animals. And then they get sick, not because of the treatment, but, just because of viral infections and whatnot. So you really, it's not fun most of the time, I have to confess. And it also made me appreciate something when we're doing these, animal models, and they really have to be, that as a scientist, that it's just animal models, large animals, it's really hard. But it's another level. I wasn't expecting it to be this hard. And, when you're reviewing papers, right, for large animal models, you really need to understand how hard it is before you ask for a new arm of a completely different, like that wasn't the question, like maybe you want to do that study, write a grant for that. That's not what we're doing here, right? We have this question and we can only really have to use this money, which is super expensive, right? It's about like $50,000 per animal. So, really making sure it's consistent and reproducible, so you can, draw any conclusions at all. If you have 50 different conditions, that's it. You're never going to be able to draw any conclusions, right?
I sense that reviewer number 3 was perhaps a little bit rough, Sarah, but or not.
Not submitted yet. We haven't submitted yet.
Okay.
I already know what's coming,
Oh, for sure. I mean, you don't want somebody like me who's an in vitro biologist looking at a large animal study and trying to do those analyses and telling you to do X number more animals. I perhaps don't appreciate how complex and how expensive these things are, but you most certainly do. You were alluding to, of course, the long range of studies that have been done in this field, focusing on cell therapy and cardiomyocyte immaturity. I mean, Chuck Murray, we've talked about a lot of his work recently. Although they're starting to move towards, perhaps addressing some of those concerns of arrhythmias through more mature cardiomyocyte transplantation, in vivo maturation, so on. But regardless, like what you're talking about, there are a lot of moving parts here, a lot of variables. The animals can randomly get sick. At times, I'm very happy that I'm just an in vitro biologist, but at the same time, these studies are very high profile, right? Ultimately, they do lead to big papers, so that's great. I actually have a separate question focused more on the engineering side of things, more on the ex vivo side of things, and this is something that comes up occasionally here on the show. I mean, we talked about how the blood vessel network and the vasculature is so intricate. There's so many different types of endothelial cells, vascular cell types, right? There's that statistic where if you put all the blood vessels in your body next to each other in a straight line, they would stretch to the moon or something like that, right? I hear about that a lot. And as a cardiovascular tissue engineer, do you think it's going to be possible to actually fully engineer that level of fine complexity and branching morphology ex vivo all the way down to that capillary level using maybe like, I don't know, bioprinting, 3D bioprinting? There have been examples of 3D bioprinted trachea and bladder, some of these really simple tissue structures, but do you think we're ever going to get there for vasculature, especially down to like the capillary level?
I think the system needs room for self-calibration. So what I think I'm trying to say here is that you can do it for the larger vessels, right? But when it comes to the smaller vessels, there is this intrinsic program. If a vessel is redundant, it needs to be cut back, right? If its oxygen levels are not good, it needs to grow. And I find that some approaches in engineering can be very hard on this tube is going to be a vessel, and as much modeling you can do to kind of distribute them evenly in a way that mimics, native tissues, that may not necessarily be the best, right? That said, I think there's a lot of room for progress. I think we can do a combination of approaches like 3D bioprinting with microvessels and at different scales to be able to do some of this work and increase intricacy. Now, how perfect that's going to be remains to be determined. We're actually in collaboration with some people that do 3D bioprinting, and they have some grants, like large team grants that we're part of, that are trying some of these approaches, right? A 3D bioprinting in combination with different strategies. So I think there's room for improvement. You have to be smart about it, and I think key is not try to control absolutely everything. Where the boundary is, right? Then it's to be determined how much is too much and how much is too little. But that has to be done experimentally, trial and error, and figuring out how it's going to turn out. I think we can increase complexity somewhat, and then the question becomes why, right? So are we doing this to put it in vivo and make things regenerate the tissue, or are we trying to do some of that stuff for in vitro application, which in that case, do we even need it, right? How simple can we go to still be able to address some of those questions?
Yes, the age-old question, we come back around to it, build it or grow it. And I think the answer is always, yeah, we're going to have to do both. And we're going to have to count on that, deep knowledge of the cell. Now, I mean, our understanding of the cell pales in comparison to the ability for the cell to understand its surroundings and react. And I guess what we've tried to do is to recapitulate that, but I think that's a heavy lift. You know, Mother Nature's had probably what, hundreds of millions of years to tune that up. So we can take a whack at it, but I think as you're saying there, we can reach some level of sophistication, and ultimately the why is huge. I can remember a decellularization, right? That was the key a while back. I was like, oh, we're gonna have to get vasculature. We'll decellularize it and then seed it back. But like, what was the end point there? We wanted to grow whole organs, and maybe realize that that was a bit more far-fetched. So yeah, the intent, the end point is critical to the effort. And I think we're doing pretty well as is, just trying to understand how these cells tick and how they play poorly in disease. And a lot of that is due to these models that have gotten pretty sophisticated.
Yeah, and I think that's why it's also important to highlight that we do a lot of like translational oriented research, but the basic part of it, the fundamental knowledge, is also important, right? We can't forget about that. So those go hand in hand. The more we know, you know, the easier it is for us. And sometimes, you know, people are very critical of like, why do we want to know? Who cares? Like, how can you apply this? I disagree, right? I think, you know, curiosity in research is really important, and just trying to figure out how it works is fantastic. You don't need to apply it for something, at least not yet, right? But in the future, you're going to realize, oh, that was the key, right?
Yes, nothing like the pure search for knowledge. I mean, we just covered a story on turtle organoids in the last episode. We love that kind of stuff on the show. And we love what you've come to share with us as well. Thank you so much, Sarah. Before we let you go, we got a couple quick science peripheral questions. First being, if you were not a scientist, what do you think you'd be?
Well, so a clarifying question. Could I be anything independent of my skills?
Anything.
So I think I would be a ballet dancer. I think that is totally out of science. But it's so much fun, you know? You can dance, the music, you're just in your head. You're not thinking about anything else. Love it.
That's a great answer. Another one for the books. Catch you in the Nutcracker. I bet you do pretty well with that. Finally, what is the biggest misconception about science that you would like to resolve?
So I don't know if that's the biggest misconceptions, right? But a lot of people I talk to think the scientists are extremely smart or super socially awkward, or right? And that's if you don't fit the profile, you're not. And that's not the case. If you've ever been to a conference or a department, you're going to see the whole spectrum, right? As you would see in any profession, I think. So really, you know, it's a bunch of smart people, right? But very curious people working, you know, together to really advance the field, because, you know, it's the passion, I think, that comes across when you talk to different scientists. So it's not, you know, just the stereotype of the, you know, crazy scientist. It's really, you know, it's actually very cool people. If you go to a conference, you have lots of fun just chatting about very nerdy things, but, you know, it's actually, it's light, right? And it's, anyway.
Yes, 100%. The best thing about this show is getting to meet all these scientists of all types. I mean, we got a nascent ballet dancer here with us. We talked to a guy the other day, has a rock band. I mean, really all types and cool people. I have to say, it can be some social awkwardness. I'm not going to lie. There's some socially awkward moments. Amongst myself, I go to a party and I'm cringing at the things that come out of my mouth. But yeah, it's really a fun time. I mean, Arun and I also talk about it when the one common denominator, as you alluded to there, is that you get a bunch of scientists together and they just start geeking out about whatever it is. And that's the most fun thing ever. So you non-scientists out there, get involved. You know, you got to be passionate about something. I'm sure the scientists get on the same page. You'll find that we're pretty cool people. And from, a socially awkward geek to a cool person. Sara, I say thank you for showing us the way with the modeling, with the regenerative. I mean, all facets. You really cover the bases when it comes to the expansive field of vascular biology. And we appreciate you sharing a little bit of everything with us.
Thank you so much for having me.
All right, you guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. Thank you so much for listening to this episode. Join us again in a couple of weeks where we'll have a fresh one for you.